Welcome to CR Arthritis 2025. My name is Ellen. I am the Programs Coordinator at Arthritis Consumer Experts. Today, I am joined by Lamia Khan, who is a PhD candidate. Lamia will be presenting Metabolic Stress Remodeling, Insights into the Role of the FOXO1 in Promoting Cancer-like Changes in Systemic Sclerosis. And Lamia, you correct me if I totally butchered that. But Lamia is at the University of Alberta. Her supervisor is Dr. Mohammed Osman, and she's recently received the Ballerman Translational Research Fellowship Award for her work. So we're really grateful to have you, and we're really excited to dive in. What is metabolic stress remodeling? So thank you so much for joining us, Lamia.
Hey, thank you so much for having me. I'm glad that I have the opportunity to present here today and talk about my research. I'm very glad, and thank you so much for inviting me.
Thank you, Lamia. Let's start by asking you, tell us a little bit about yourself and how you're involved in rheumatology. So get into a little more specific about your research, if you don't mind.
Yeah, sure. I'm a researcher, obviously, and I'm studying my PhD at the University of Alberta, as you mentioned, give a nice introduction, and it's in the Department of Medicine. And my supervisor is Dr. Mohammed Osman, who is a clinician scientist, and also he's the director of SSC Clinic here in Alberta. And my PhD research is on systemic sclerosis. Also, we call it SSC or scleroderma. And it is a serious autoimmune disease that causes abnormal thickening of the skin. The term scientific, we call it fibrosis, and it happens in the skin and organs. And this thickening of the skin causes the life-threatening complications, which can eventually lead to death. And we recently discovered that patients with severe systemic sclerosis, they develop DNA breaks in their skin cells, which can lead to harmful mutations like cancer. And now we are investigating how these DNA breaks regulate metabolic stress signals to drive this abnormal skin thickening. And we hope that if we can understand this process and decipher it, there will be a new ways to slow down or delay the disease progression, or stop it and improve treatments.
Incredible work, very much so cutting edge. But let me repeat back to you what I understood. So you work specifically on a condition called systemic sclerosis, or
Correct.
also known as scleroderma.
Yeah.
And it is a condition where there is fibrosis or damage to the DNA of your skin cells. So when they replicate, right? Because everyone gets dry skin, that's because your skin cells replicate.
The normally happens, the replication, right? But in this process, DNA can, like DNA breaks, but it can be permanent, and which is like scary. It should not happen. It persists longer, and that is happening in systemic sclerosis patients.
I see. And as you said, because of the DNA damage and the fibrosis and the ongoing, right? It's not once, keeps on happening. There may be complications medically. So you are looking specifically at detecting these DNA damage that happens in the skin so you can catch the disease earlier on, because we know, we can catch the systemic sclerosis as well as cancer, because the DNA damage can also lead to cancer. Am I understanding that?
Yeah, and it's the cancer-like changes. We see properties in the systemic sclerosis skin cells that they have cancer-like properties, like they survive longer, not like the healthy cells under the stress condition, or even if we induce cell that they don't die. So which is a cancer-like properties also.
Cancer-like properties. Thank you for clarifying that. So in that case, Lamia, what are some of the key takeaways from your message and from your research, I guess, that you're presenting at the CRA conference?
So the take-home message would be that in severe systemic sclerosis patients, they have DNA breaks in their skin cells, termed fibroblast, and which causes the activation of a molecule, stress response molecules, FOXO1, which is a protein and it's a transcription factor that regulates gene expression in our body. And and this activation of FOX1, it causes the alteration in metabolism. It causes the mitochondrial dysfunction. Then which causes the increase in the oxidative stress, or increase the cellular oxidative stress. And these fibroblasts or skin cells, they live longer, leading to excessive scarring of skin.
Wow, very interesting. So you've narrowed down, your research is able to narrow down the fibrosis causing molecule called FOXO1. So for
Yeah.
everyone, it's literally FOXO1 is what they've named this molecule, right? As Lamia had told us, this is a transcription factor, which means it helps to replicate DNA. Am I correct?
It helps the gene expression. So we
Gene
know,
expression.
yeah, because we know like proteins play a role in the cells, but protein expressions, they're regulated. Their expression happens in a particular situation, right? Like maybe cells need some stress response molecules. They need to turn on, and FOXO1 turns on the expression of those
And is
molecules.
it too active? Is FOXO1 too active in systemic sclerosis, or is it just not doing a good job? If you had to simplify, of course.
Okay, so let's start with the healthy cells. That it helps cells to adapt the stress response, but in disease condition, this FOXO1, they are hyperactive, which
Okay,
they should not be.
there we
And
go.
yeah, and that is actually causing the metabolic stress remodeling and causing the fibrosis in these patients. That's what our data suggests.
Excellent. So in that case, now we know that FOXO1, right, helps us handle stress. But in the case of systemic sclerosis, it's a little too active, which causes fibrosis, right, or thickening, right, of the skin cells. And of course, as you noted, those complications later down the road. So thank you, Lamia. That was so well
Yeah,
spoken.
because like we need something like a short time, it's good, but if it's happened chronically, it's persist, it's not good for our cellular mechanism. It just creates this abnormal environment, or it alters like the cellular environment.
And I think that's a really key point. When everyone thinks inflammation now, they think if all inflammation is bad, which is untrue because if you're sick or you have a virus or you got a cut because you fell down, there's
Mm.
some level of inflammation that's good. There's some level of fibrosis that's good, right? Adaptive, like you had noted. But the issue is when it continues on and it doesn't turn off, as you noted, those complications. Yes.
And we
So,
found it. We found like, so we study the skin cells,
mhm.
fibroblasts. So my supervisor, so he takes the skin samples from the patients, systemic sclerosis patients, and we use these cells to grow fibroblasts. That is the like skin cells, and also a key driver of the skin thickening process in these patients. And in these cells, we saw that there's a higher activation of this FOXO1. That means the FOXO1 is basically, it remains in the cytoplasm in phosphorylated form, and then it gets activated and comes to nucleus and retains there, because in nucleus, all our genes are there, drives the expression of those genes. We found like higher, very increased levels of FOXO1 in the systemic sclerosis patients' skin cells.
Very interesting. Like you had said. So this FOXO1 usually hangs out until it's needed, right?
Yeah,
In this case, they were doing too much work, and they were, like you said, overactive.
yeah, overactive.
Overactive. Very, very interesting. And personal question for you, this year's meeting, right? The theme is Ascending to New Heights, Peaks of Innovation in Rheumatology. What
Yeah.
are, Lamia, what are you looking forward to? What are the latest innovations that you personally are really excited for? It can be in your research, it can be your lab research, it can be in rheumatology more broadly that you're looking forward to.
Yeah. So, like we mentioned, we are studying how skin cells behave abnormally in systemic sclerosis, right? The fibroblast, how it behaves, and we mostly do the functional studies. But we are very excited to include like the single cell multiomics approach and the spatial transcriptomics with our study, because if we can combine this advanced what I should say innovative approach with our functional studies, then it will give us a broader picture how this fibrosis is happening at molecular and cellular level. We'll see how cells are interacting with each other to drive the disease progression. And in our research, we have already started implementing this, because these are very powerful techniques. And then we are using the spatial transcriptomics, and then also we found some changes in the mitochondrial signature in systemic sclerosis patients' skin with this approach. So, and we want to more integrate this approach to our research. Yeah. Another thing I would like to mention. So we also work with the autologous stem cell transplantation.
Mhm.
So for systemic sclerosis patients, there are very limited treatment options. But so far, the most effective disease-modifying therapy is this autologous stem cell transplantation. So we are also, so it is speculated that autologous stem cell transplantation, it reboost or reset immune cell function in these patients, and thus improves it. But also this shows that this treatment improves the skin fibrosis or thicken skin in the patients, but it is just not known how how this treatment affects particularly the skin cells. So we are also like studying the fibroblast or skin cells from these patients to understand this mechanism, and we are very hopeful about it, that if we can understand that, we also can like it can help to develop new treatment strategies.
Like you said, like systemic sclerosis is more rare, and there's been so little research on it. So the fact that, your team is dedicated to advancing this field, and it seems like there are so many things that we haven't tried yet, right? Like we haven't looked into to gain that broader understanding, as you had noted. And I'm very curious of what is the next step with this skin stem cell transplant, and for developing new treatments for autoimmune conditions, generally speaking. And yeah, it's just a very interesting time to be in the field.
Exactly. We want to see where it will take us in near future.
Very near future.
Yeah.
With that, do you have any other insights you want to share to patients about their disease journey, self-care, self-advocacy for a patient? For example, I'm someone living with systemic sclerosis, and as you had noted, limited treatment options. What advice or suggestions would you give this individual?
Exactly. So I'll just share, I had some opportunity to meet with some of the patients in the clinic of my supervisor, Dr. Osman. So I saw like living with systemic sclerosis is not easy. It's very challenging for them, and it affects like various aspects of life. So obviously they need that support, the therapy and mindfulness, which can make a big difference, right? And also I think like we should like as a researcher, as a clinician, as a rheumatologist, we should make them educate or like informed about the recent advances, like what the research is going on in this field, what recent treatments are available for them, pros and cons. They should know about this, because knowledge is power, and it is the disease they're surviving with. They should learn about it, and I think it will help them to choose their therapies. They will understand it. They can also improve their lifestyle, or like adjust their lifestyle depending on that. Yeah.
I agree. There's so many, like, obviously you work in the field of developing new treatments and understanding the disease itself, but the concept of care is more than just developing the right medications, right? Like
Exactly.
the other components around the individual, social support, how to support them emotionally. There's so much work to be done there. So, Lamia, thank you so much for all of your insights. Your work is so interesting and so incredible. I am very excited as you progress through your PhD. You'll keep us informed. And with that, thank you for being here and sharing your time with us.
Thank you so much, Helen, for having me, for giving a nice introduction, for keeping me lively throughout this interview or like throughout this conversation. I really enjoyed it. Thank you so much.
Thank you. And with that, thank you to our audience for tuning in. Make sure you check out our other episodes from the CR Arthritis event 2025. Lots of fantastic interviews with different researchers, trainees, clinicians. And with that, take care, everyone. Have a great rest of your week.
Yeah, thank you so much to all our patients and all our audience. Thank you.